Hello students, in the last lecture we have discussed with the differences of prokaryotic and eukaryotic cells, and today I will be discussing with the eukaryotic cell. We will be starting with the eukaryotic cell. In that I will be first discussing with the cell wall. Cell wall it is seen in both prokaryotic as well as the eukaryotic organisms. We have discussed this in the differences. There I have told you that the prokaryotic cell, mainly the bacteria, they are made up of peptidoglycan. And what about the eukaryotic organisms? We have four types of organisms: plants, protista, fungi and animals. Out of these four, animals completely lack the cell wall. What about the other three organisms? The fungi, they are made up of the chitin, it's a different kind of polymer, and we have cellulose which is present in the plants, and also in the protista we have the cellulosic cell wall. Majority of the protistants, they lack the cell wall. If the cell wall is present, they will be having the cellulosic cell wall. And coming to the cell wall of the plants. So cell wall today we'll be discussing with the plant cell wall. All the plants, they have the cell wall with few exceptions. There are certain lower group of algae where the gametic state, they lack the cell wall. So the lower organisms, the algae, the reproductive phase will form certain gametes. We all know during the reproductive phase, the gametic formation will be taking place. So these gametes, they lack the cell wall. This is one of the exceptions. All the plants, they have cell wall with one or a few exceptions. You can say that is in the lower group of plants, algae, the gametic stage, they lack the cell wall. This is one exception where you don't see the cell wall. And for the first time, the cell wall has been discovered by the scientist Robert Hooke. Cell as well as the cell wall, both of them, they were discovered by the scientist Robert Hooke. Coming to the cell wall and its structure. How the cell wall important for the plant life? Cell wall, it is the external outermost layer of the plant cell. So it is the outermost layer of the plant cell, and it is present above the plasma membrane. We all know the cytoplasm, it is bounded by a plasma membrane. Above the plasma membrane, we have this cell wall. Then what is the function of the cell wall? The first important function it will determine the shape and structure of the cell. Then what is the second important function? It provides protection, security to the plant body. How this is providing security? The cell wall plays a key role in the life of the plant body. It provides a high-class security to the plant body. So here, in the case of the plants, we all know the plants are immobile in nature. They can't move. They have to survive in the unfavorable conditions which are present in the environment, children, right? What are the different type of conditions? We have high wind, or you can say humidity, or temperature, low temperature, high temperature. So all these different type of conditions are unfavorable for an organism to survive. So if you take the case of animals or human beings, yes, we can escape the unfavorable condition, but can they escape these conditions, children? No, they cannot. Why? Because they are immobile, they are static. As they are static, as they have to face all these limitless challenges present inside the environment, they require an extra protection. And this extra protection, it is given to the plant body in the name of the cell wall. Okay? And not only that, the plant cell wall, it is highly impermeable to different type of substances. It hardly or it rarely allows the substances to pass inside unless it is wounded. And if you compare this with the higher organisms, the plants even lack the immunity. A proper immune system or immunity, it is lacking inside the plant body. So in order to balance and manage all these functions, prevent all these conditions, the plants will be having this cell wall. So we all know the prevention is better than cure. So the same principle is applied here. It is having a preventive measure to help the plants to survive and sustain in the environmental conditions. Okay, so this is regarding the functions of the cell wall. So these are the two major functions. One is it is helping in determining the shape and structure of the cell, and the second it is providing the protection to the plant body. And next, coming to the components of the cell wall, how the cell wall is present, that we will see. General view of the cell wall observed under microscope. Next is the structure of the cell wall. The structure of the cell wall, it is mainly studied under three major components or three major headings. What are the three major headings? We have the matrix, second is microfibrils, and third is the deposition. So what is the matrix here? Matrix is the gel-like substance, or you can say it is a gelatinous substance which is present in the cell wall. And what does this gelatinous substance contain? This gelatinous substance contain, if you observe the diagram, look at the structure, children. Pectin, we have proteins, we have hemicellulose, will be present inside the cell wall, inside this gelatinous matrix. Then what about the microfibrils? Microfibrils are these rod-like structure. So only these rods, they constitute the microfibrils. So the second region is nothing but the rod-like structures. Okay. And the third one, it is not shown here, children. These are depositions. Sometimes the plant cell wall, they will have the extra deposits present over it, and these are known as depositions. They may be present or absent. Based upon the condition, the plants will show certain depositions. So we'll be studying all these three structures. The first I have told you, the matrix, which is the ground substance. It contains all other structures except these rod-like structures. And these rod-like structures, they constitute the microfibrils. And we have depositions. I have not shown any depositions here. Depositions means these are extra deposits which are present above the cell wall, and based upon the nature of the plant body, they will show these extra deposits that we will be discussing. Okay, so first we will start with the matrix. Matrix is gel-like ground substance. So this is gel-like ground substance. So here we have different type of structures present in the matrix. Mainly this matrix, it is made up of 30 to 60% of water. So water will occupy the major portion. So here water, it is 30 to 60%. Okay. Then what this matrix it is made up of, other than water, what we have inside. So after this, we have the second important substance, which is the hemicellulose. The majority of the matrix, it is having water. Okay, that is 30 to 60%. And next we have the hemicellulose. What is hemicellulose? Hemicellulose, it is carbohydrate. We all know it's a sugar compound. And in sugars, we have two different type of sugars, I have told you. What are the two different type of sugars, children? We have homopolysaccharides and heteropolysaccharides. What are homopolysaccharides? If the particular complex carbohydrate, it is made up of a same type of sugar, we call it as homopolysaccharide. One example I will be giving, one is cellulose. Cellulose, it is made up of many sugar units, and all the sugar units are the same. It is made up of glucose sugar units. Many glucose units, they will bind to form a complex carbohydrate, that is the cellulose. And in the case of heteropolysaccharide, different type of sugars, they will bind together to form the complex carbohydrate. So one type of carbohydrate which will be formed with the different sugars or combination of sugars, we call them as heteropolysaccharide. Then what is hemicellulose? It is a heteropolysaccharide. Hemicellulose is heteropolysaccharide. What is the nature of hemicellulose? It is not soluble in water, children. It is insoluble in water, and it is short and branched. It is short branched heteropolysaccharide which is insoluble in water. How much amount of hemicellulose is seen? If you observe the picture, I have given these orange colored structures, and all these orange colored structure, they represent hemicellulose. So hemicellulose, it is present here. Okay, and what is the hemicellulose percentage? I have told you it is around 5 to 15%. So it occupies 5 to 15%. Then what I have told you, that this hemicellulose, it is a heteropolysaccharide. Then it will be having different type of sugar units. What type of sugars are seen in the case of this hemicellulose? That we will write now. So what are the different type of sugar units that are present in the hemicellulose? In order to form the hemicellulose, we have different combination of sugars. The sugars can be galactose, mannose, arabinose, xylose and uronic acid. So all these different type of sugars are forming the heteropolysaccharide compound, that is hemicellulose. Sometimes this hemicellulose are also said to be having a different type of compounds, which are arabinoxylon and galactomannose. What are these compounds? Here xylon means many xylose units, they form xylon, and they are combined with one arabinose or arabinose sugar, and they will form xylon arabinose combination, that is the polysaccharide. And in the same way we have galactose and mannose. What are mannose? Mannose means mannose units, many mannose sugars, they will form one mannose. Okay, and here these mannose will be binding with the galactose to form galactomannose. So these are combination of two type of sugars, galactose and mannose. Arabinoxylon means here the xylose sugar, it is combined with arabinose sugar, and they will form a combination carbohydrate which is known as arabinoxylon. Here xylon means xylose units, many xylose sugars, they will bind together to form a xylon. And here again I have given galactomannose. Galactose sugar and mannose sugar, they will combine to form galactomannose. But here mainly they have mannose, means mannose sugar units will be more compared to that of the galactose. So many mannose sugars, they will combine with galactose to form galactomannose. So such type of compounds are seen in the case of hemicellulose. And next, coming to the third important component of the matrix, that is the pectin. Pectin will occupy 2 to 8%. So it will be occupying almost 2 to 8% of the volume in the matrix. What type of substance is pectin? Pectin is again a heteropolysaccharide, but it is soluble in water. Unlike that of the hemicellulose, which is insoluble in water, pectin is highly hydrated compound and it is soluble in water. And again, the pectin, it is mainly forming rigid gels. With the ability to hydrate, it will form rigid gels. Pectins will be mainly having high negative charge. Pectins will have high negative charge mainly because of the sugars which are present in them. Those are highly negative in nature. Okay. So here a pectin compound, the pectin carbohydrate, it's a heteropolysaccharide. It is mainly made up of two types of sugars. What are the two types of sugars that are seen in the pectins, children? Galactouronic acid and glucuronic acid. Both these type of acids, they are mainly having high negative charge. So pectin will be having, the total charge of the pectin will be negative. Why? Because they are mainly made up of two type of sugars which are imparting a negative charge to the pectin molecules. Because of this nature, because of the highly negative nature, what does pectins will do? The pectins will be affinity with the cations. As they are having negative charge, they will bind with the cations. Cations like magnesium and calcium. And what they will do? In turn, these pectins will be binding with the cations like calcium and magnesium, and they will form semi-rigid gels. Okay, due to their hydration ability and also the ability to bind the cations, they will form the semi-rigid gels. If you remember, I have told you that in the middle lamella, while we are discussing with the differences in prokaryotic and eukaryotic cells, I have told you the plant cells will have the middle lamella, which is a cementing substance, and the cementing substance, it is present in between the neighboring cells of the plant. And here this middle lamella, it is made up of calcium and magnesium pectate. So how these are formed? Now you can understand. That is mainly because of the ability of the pectin to attract the water molecules and attract the cations, that is calcium and magnesium. So they will be forming the semi-rigid gels. Okay, and this is a major component of the middle lamella. You must understand that the pectins are highly hydrated molecules. Not only that, they have the negative charged sugars, that is galactouronic acid and glucuronic acid. Because of this negative charge, they will attract the cationic molecules or cations, which are calcium and magnesium. So they will in turn form calcium and magnesium pectate, which is a component of middle lamella. So next coming to the third component, that is the lipids. Lipids are the fat molecules, and these lipids will constitute 0.5 to 3% in the matrix region. So in the matrix region we have 0.5 to 3% of the lipids. And finally we have the protein. The proteins will be around 1 to 2%. So glycoproteins, they will be increasing the mechanical strength of the cell wall, and they will constitute 1 to 2% of the total cell wall matrix. Okay. So this is regarding the matrix. So matrix region of the cell wall, it is made up of 4 major biomolecules. What are the 4 major biomolecules? The first we have the water. Water will occupy 30 to 60%, and next we have hemicellulose. Hemicellulose will be having 5 to 15%, and third we have pectin, and the pectin will constitute 2 to 8%. And finally we have lipids and proteins. Lipids will occupy 0.5 to 3%, and finally the proteins will be constituting 1 to 2% of the matrix in the cell wall. Okay, so this is regarding the matrix. And after the matrix we have the second one that I have told you, microfibrils. So this ground substance we have discussed here, children. So what have we discussed? One, I have told you water, and the second I have told you hemicellulose. If you observe the diagram, these are hemicellulose molecules. And next we have discussed with the pectin, lipids and proteins. These are protein molecules. This blue colored helical chains are proteins, as well as the red colored ones, both are protein chains. And next we have the lipids and pectin. These green colored ones are the pectin, and we also have certain lipid molecules which are attaching with the cell wall. Okay, matrix region. And next we have the second component, that is the microfibrils. Here the microfibrils are nothing but the rod-like structures or cylindrical structures. They are almost parallelly arranged in the cell wall. Sometimes they may differ in their shape based upon the age of the plant. Okay, and here if you observe, mainly the primary cell walls or the immature cells or young cells, they will have this structure. These microfibrils are rod-like structures. They are arranged parallelly, and they are mainly made up of a homopolysaccharide, that is the cellulose, a straight chain molecule. And I have told you that it is the same type of sugar units that will form this cellulose. Which type of sugar is present in the cellulose? Glucose units. So many glucose sugars, they will bind together, they combine together, and they will form the cellulose molecule. This is not branched compound like pectin or hemicellulose. It is a straight chain molecule. Okay, so it's a straight chain compound. So in between these straight chain molecules, again we have many hydrogen bonds, and they will form a rod-like structure like this. So they will form a rod, like many straight chains are arranged, and they are again interlinked with the hydrogen bonding, and finally they are forming or giving out a cylindrical structures or fiber-like structures. These are known as the microfibrils. In the plants mainly we have the cellulose, but in the case of different organisms, the fibrils are made up of different chemical composition. Let's discuss. So one is being fungi. So in fungi, I have told you they are mainly made up of a homopolysaccharide which is known as a chitin. Okay, and we have yeast, bacteria and certain fungi or fungal species. They will be having mannans. Mannans means they are again homopolysaccharides. They are made up of mannose sugar. And we have another example in the case of algae. In the case of seaweeds, we have agar-agar, that is agar gel, and agar gel, it is mainly made up of the galactose unit. So these are the different type of compounds that are present in the cell wall of the different organisms. Okay, so first I have told you that the plants will have cellulose, and we have fungi. Mainly the fungi, the cell wall is made up of chitin. And in certain fungi, they will also be having a different compound which is made up of mannose, and these mannose units, many mannose units, they will form the mannans. Bacteria are also showing some mannans. And next we have seaweeds. Seaweeds are showing agar-agar. Agar-agar, it's again a polysaccharide or a sugar compound, and it is mainly made up of which sugar, children? It is made up of galactose. So these are certain examples where the microfibrils will be having different chemical composition other than the cellulose. And next coming to the third part, that is the depositions. And next coming to the third component, that is the depositions. The third most important component of the cell wall is the deposition. So we have to discuss the depositions. I have told you they may be present or absent. They are the extra deposits that are formed on the cell wall. Based upon the age and functioning of the plant cell, the cell wall will be showing different type of depositions. The first being the lignin. This lignin, it is a kind of hydrophobic polyester compound, or you can say polymeric compound. Lignin, it is a kind of hydrophobic polymeric compound. They are made up of aromatic phenols, and this is water hating substance. Okay, and this kind of substance, when it is deposited on the cell wall, it will make the cell wall impermeable. Not only that, when the lignification, the process of lignin deposition is taking place on the cell, it will make the cell the dead cell. Okay, so the dead cells are mainly because of the deposition of the polymeric compound, which is also known as a bioplastic. Okay, it is non-fibrous in nature, it is known as bioplastic, and when this lignification is taking place, it will make the cell dead cell. Okay, this is one of the kind of deposition that we will see in the plant cell. So where you can see this lignin, mainly in the case of xylem vessels, the tubes, the conducting tubes present inside the plant body, they are mainly deposited with the lignin. It will give mechanical strength as well as stiffness to the plant body. So it is mainly seen in the case of xylem. And next we have callose. Callose, it's a kind of a fatty acid molecule, and these fatty acid molecules, it is mainly seen in the case of pollen tubes as well as the xylem. Next we have the suberin. Suberin is also a kind of fatty acid compound. Okay, these are also fatty acid molecules. An example is cork. We all know the bark, external layer of the plant body, the woody tissue, the core, it will be having this suberin. Okay. And again we have the Casparian strips of the plant tissues. We have certain plant tissues where we have the strip-like structures, or the suberin, it is coated on the walls of certain tissues. Casparian strips are mainly seen in the case of certain plant tissues. We will see the functions of Casparian strips in the anatomy part. Okay, so you just remember that the suberin is present in the Casparian bands of certain plant tissues. And next is the cutin. Cutin is also a waxy substance. It is also a fatty acid molecule. It is resistant to water. These are mainly coated on the walls of the epidermis. Epidermis is the outer layer. In the leaves or stem, we have epidermal layer, and this epidermal layer is the outermost layer. On the epidermis, mainly on the epidermal cells of the leaves, these cutin, it is mainly deposited. So what is the function of this cutin, children? They mainly prevent they will check the transpiration rate. The transpiration will be controlled with the help of this waxy coating present above the epidermal cells. Okay, so it is also. And lastly we have the mineral deposits. Certain mineral or inorganic ions are also deposited on the cell walls. For example, we have the mineral deposits. In the mineral deposits, I have told you that inorganic ions are mainly present, and these inorganic ions are found as deposits on the cell wall. In certain plants like cucurbits, mainly the ridge gourd, bottle gourd, we have different type of cucurbits, and all these cucurbits, they mainly show the calcium and magnesium carbonates which are present on their cell wall. Other than that, we have an algal member which is Cara. Cara also show these calcium, mainly iron. Iron, it is seen in the case of Cara. Silica deposits are mainly seen in the case of the grasses. So this is regarding the third most important component, that is deposition. Now let us see the structure of the cell wall. So now we have seen how the cell wall it is made up of, what are the components of the cell wall that we have studied. Now we will be discussing with the structure of the cell wall, how the cell wall is formed, how many layers are seen in the cell wall. So here mainly the cell wall, it is divided into three main layers. What are the three main layers, children? We have primary cell wall, secondary cell wall, and the tertiary cell wall. So these are the three main layers of the cell wall. Other than this, we have another substance which is known as middle lamella. What is this middle lamella? I have told you that middle lamella, it is a cementing substance, and this is mainly helping in the binding of the neighboring cells. So neighboring plant cells, they will be binding together with a cementing substance, and this cementing substance is known as middle lamella. So these are the main layers. Mainly we have 3 layers. Other than this, we have the middle lamella. So what is middle lamella? Middle lamella, it is mainly the cementing substance between the cells. Now let us see. Let us say this is cell number 1. Okay. And this is cell number 2. Right? So this is one cell and we have another cell. Okay? Cell number 1 and cell number 2. So this cell number 1, we have layers here, and these layers I have depicted, I have showed in this diagram. And again with the cell number 2, here we have the middle lamella. And after that middle lamella, we have again the cell wall layers. I have shown the cell wall layers. Okay. So what, only if you see in the diagram, we have only one layer, children, but it is not actually the one layer. When you observe under microscope, we have different layers present in the cell wall. This is cell number 1 and this is cell number 2. And joining these two cells, we have middle lamella, that is a cementing substance. This middle lamella, it will be binding the cells together. Okay. So first we will see the middle lamella. Middle lamella is a cementing substance, and this is made up of calcium and magnesium pectate. As discussed before, I have told you that middle lamella, it is made up of pectin, and this pectin in turn, it will combine with the inorganic ions magnesium and calcium. They will form the semi-rigid gel, and the semi-rigid gel, gelatinous matrix, this will bind the cells together. This is the first layer that is formed during the division. During the cell division, the new cells are being formed, and in this at this time, these new cells will show the middle lamella as the first and foremost layer. The first and foremost layer that is formed during cell division is the middle lamella. And this middle lamella, it is jointed to the primary wall of the both cells. So how this is being attached, children? This middle lamella, it is adjacently attached to the first layer of the plant cell, which is the primary wall. So what is the first layer of the plant cells? Primary wall. Look at this. So let us say this is the primary wall. Okay, primary wall. And this is for the cell number 1. What about the cell number 2? We have another primary wall. Okay, so this is primary wall here. Then what is the second layer? I have told you secondary wall. Secondary wall, it is like this. Okay, this is secondary wall. Here for cell number 2, we have secondary wall. Okay. And what is the third layer? We'll mention this. These two constitute secondary wall. Okay. And the last one, it is tertiary wall. Let us dot this. Okay. So this is a structure here. After the middle lamella, we have the primary wall. On both sides, we have two cells. This is cell number 2 and this is cell number 1. The first layer of cell number 1 and 2 is the primary wall. This primary wall is the only layer which is seen in the case of young cells or immature cells. When the cell is very young, the cell wall will be having only one layer. What is that layer? Along with the middle lamella, they have the primary wall. And when the age of the plant, when the plant is growing, when the age is increased, the subsequent layers are formed. What are the subsequent layers? We have the secondary cell wall and the tertiary cell wall. Tertiary cell wall, it is rarely seen, children. Only in certain plants the tertiary wall is seen. Otherwise, we have primary wall and the secondary wall. Okay. And here, if you observe the number, the primary wall is always, it is only in a single layer. It is present in a single layer. It doesn't have sub layers. But in the case of secondary wall, you have more than 3 layers. I have given 2 layers of secondary wall on either side, but we can have more than 2 to 3 layers can be formed, that is of the secondary wall. And tertiary wall, it is rarely formed. Now let us see the differences here. So in the case of primary wall, I have told you that this is only single layer and no sublayers are present. Not only that, here the amount of cellulose will be less. I have told you that these layers, they are mainly made up of three substances. What are the three substances we have discussed? One is matrix, other are fibrils and depositions. All these three components will form every layer. But there will be differences. The components will be having, the composition of the components will be different for each layer, which will make the difference. Okay. So here, if you observe for the primary cell wall, the cellulose amount will be less and it is around 20%. Okay. So primary wall, we have 20% of cellulose. Then what is more here? What is more? Here we have the hemicellulose, pectin, that will be more here. Okay. What will be more here, children? We have hemicellulose, we have pectin, and we have lipids. So here we have hemicellulose, pectin, lipids and proteins, everything it will be more. The hydration ability will be more. Why? Because the pectin content is more. Whenever the pectin is high, they will absorb the water molecules and hence the hydration ability or capability will be more for the primary wall. And here the cellulose content is very less. Why? Because the cellulose microfibrils are very loose and they are wavy. Why? Because they are mainly seen in the case of immature cells, growing cells, and there the cellulose content will be less. And the most important point, the lignin, it is absent in the primary walls. Okay, so I have told you when the lignin is deposited, it will make the cells impermeable. So when it will make the cells impermeable or lignification will be taking place, it is mainly when the plant is reaching certain age, the lignin deposition will be taking place to make it strong and stiff. Okay, so in the primary wall, we doesn't see any lignin. Lignin is absent. Sublayers are absent, lignin is absent. What is more? Hemicellulose, pectin, lipids and proteins are more and the cellulose content is also very less. Okay, so this is regarding the primary wall. Then what about the secondary wall? In the secondary wall, I have told you the sublayers are present. Okay, the sublayers are present. How many sublayers? It can be from S1, S2, S3, so on, more than 5. Okay, more than 5 layers of the secondary wall can be formed in the case of the plant cells. Okay, I have shown you only 2 here. Okay, so these 2 are the secondary wall, but we can have more than 3 layers or 2 layers here. And the most important thing here is, if you compare with the primary wall, the cellulose content is very high, that is around 40%. So we have 40% of cellulose. So we have 40% of cellulose. And what about the remaining all substances, children? Hemicellulose, pectin, lipids, proteins, all these are very less. Okay? Lipids, it is comparatively less. Then what about lignin? Lignin, it is definitely formed in the secondary cell wall. Why? Because due to the increase in age of the plant cell, they will start forming these secondary layers. Okay, so secondary layers or the secondary wall, it is mainly formed in the aged plants or the plant having more age. Then what will happen? The cells will tend to show this lignification or lignin deposition will be seen. Okay, and when this lignin is present, what I have told you, the stretching ability or the flexibility of the wall will be decreased. The stiffness will be increased. The wall stiffness will be increased. As the pectin is less, I have not written here pectin. So all these components are less. When the pectin is less, the hydration ability will be less. And when the lignin is present, it will make the wall stiff and hard. Okay. So these are the major properties of the secondary wall. Compared to that of the primary wall, the secondary wall is stiff and it is also hard in nature. Sometimes the primary wall is also impermeable, though the lignin it is absent here. Certain substances called cutin, the waxy substances we have discussed in the deposition. These waxy substances are coating the primary wall. When the primary wall it is deposited with the cutin, then the primary wall it will be turned into an impermeable layer. Okay, so that's a different case. Other than that, mainly it is permeable to different kind of substances. But when the secondary layer it is formed, the cell will become impermeable. Why? Because lignin deposition is mainly seen here. Okay, and next coming to the third layer, that is the tertiary wall. Tertiary wall, it is generally not formed, children. Tertiary wall, it is seen only in the case of certain plants. And this tertiary wall, it is mainly made up of a component called xylon. So it is mainly having the xylose sugar, and hence we can easily detect these primary tertiary wall from the other two walls. How you can differentiate all these walls? Because they are having different type of staining techniques, different components and different composition of components are present, and hence we can easily declare each of them differently. The primary wall will be having different substances, the secondary wall will have different composition. In the same way, the tertiary wall, it will be mainly having a different kind of sugar, that is known as xylon. Because of the xylon, we can say that there is another layer which is formed, which is the tertiary wall. This is rarely formed and it is seen only in certain type of plant species. So tertiary wall, it is mainly having xylon and it is differentiated from the primary and the secondary wall. And another important point here, this primary wall, it is in direct contact with the middle lamella. When you observe under microscope, the layer, the middle lamella and the primary wall, it will be fused completely, showing a compound middle lamella. It will appear like a middle lamella itself. Why? Because we can't see or distinguish this primary wall. As this primary wall, it is completely fused with the middle lamella. So you cannot observe the primary wall of the either of the cells. Both the cells, you cannot see this primary wall, and you can see a compound middle lamella. You will observe a thick middle lamella which is fused with the primary wall. So when you can observe this difference, when this layer, this middle lamella, it is dissoluted, then you can clearly see this primary wall. There is an enzyme called polygalacturonase. Okay, we have an enzyme. We have an enzyme called polygalacturonase, and this is mainly helpful in the dissolution of the middle lamella. When the fruits are softened and the vegetables, when they are softened, they mainly have this middle lamella which is dissoluted completely. When you see this dissolution, you have the clear demarcation of the primary walls of each cells. Okay. Otherwise, you cannot see the primary wall separately. You can see that primary wall is completely fused with the wall of middle lamella. Okay, so this is most important, children. This middle lamella, it will be loosened when the you can see this in the case of the ripening of the fruits and vegetables. The fruits and vegetables, they will get softened because of the dissolution of the middle lamella. And what is the enzyme that is playing the role? That is polygalacturonase. It is mainly helping in the dissolution of the middle lamella. So this is regarding the structure of the cell wall. What are the components of the cell wall? There are three main components. One, I have told you, matrix. Second is the microfibers, and third is the depositions. So with that, we have layers of the plant cell. So all these three components together constitute the cell wall. And how this cell wall is present? Cell wall, it is present mainly in three layers. What are the three layers? Primary, secondary and tertiary walls. Mainly in mostly the plants will show the primary cell wall and the secondary cell wall, and rarely we see this tertiary cell wall. Okay, so this is regarding the composition and structure of the cell wall. And next we have two important terms that has to be discussed here. One is intussusception and the other is the accretion. So the growth of the primary cell wall and the secondary cell wall, it is slightly different. So based upon that, we have two important terms that has to be noted. The first one, it is the accretion. What is accretion? So during the growth of the cell, what will happen, children? The deposition of the substances or materials will be taking place. In the case of the secondary wall, so in secondary wall, let us say this is secondary wall, what will happen? The deposition of substances will be taking place on the pre-existing cell wall. If you observe, so here the deposition will take place. Let us say this is the secondary wall, and on this secondary wall we can see the depositions, and such type of process, it is known as accretion. It is known as accretion. This is seen in the secondary wall. In secondary wall, the deposition of the material or the substances will be taking place above the pre-existing material, which is known as accretion. Then what is about the primary wall? In the primary wall, let us say this is the primary wall. So this is the pre-existing primary wall. If the primary wall has to be grown and the substances has to be deposited, the substances will be depositing inside the wall. Okay. So this is above the wall. on the pre-existing wall, the wall material will be depositing. But in the case of primary wall, what will happen? Inside the wall, means inside this membrane or inside this layer, we have the deposition of the material, and this process, it is known as intersusception. So these are the most important points you have to remember. One is accretion, this is seen in the secondary wall, and we have intersusception, which is seen in the case of primary wall. Okay. So primary wall, it will show intersusception. What is intersusception? Here the layer which is formed will be present, and the deposited material will be placed within the primary wall, which we call it as intersusception. In the case of the secondary wall, what will happen? Accretion will take place. What is accretion? The deposition of the material above the pre-existing material or pre-existing layer is known as accretion. So these are the most important terms that you have to remember. So this is regarding the cell structure or the cell wall structure in the plant cells. In the next lecture, I will be discussing with the plasmodesmata.